as I told you, I have given you this introduction a few minutes back. Dr. Mort is a senior bioinformatician, and he takes care of the humanity mutation database. He's basically from technology background, but believe me, he knows biology better than anybody. At least I can vouch for it. At least he knows biology better than me, right, Matt?
No, you've been some time.
So Matt and I were like friends for many days, and we were in the same lab during my postdoc days. And Matt is a fantastic person and a fantastic teacher, because whenever I face any problem, I shift to Matt, and he simply solves it like anything. So over to you, Matt.
Thank you, Anne, for that nice presentation and introduction.
Okay. Everybody can hear you. I think it's fine.
Right, I'm just getting up to the presentation. Right, can everyone see that, Anne?
So
Can you hear me, Anne?
yeah, I can hear you.
I can't hear you now, but that's probably
Yeah, I can hear you. Yeah, sure. So I'm turning the videos off.
Perfect. Right, thank you, Anne, for that nice introduction. So I'm going to talk about primer basically on interpreting and understanding the genetic variation. So we're going to be focusing on the movement, we're going to be focusing on mainly variation causes disease. So that's what, in my current job, I work with a human gene mutation database. We basically got a database of mutations that cause disease. So that's sort of my primary interest. So I'm just trying to switch to another stuff. Right, so what is a genetic variant in humans? That's what we're going to start with now. I'm assuming everyone can hear me now. So a genetic variant is otherwise known as a mutation or a polymorphism. So you will have heard those terms used. And what do we mean by this? So we mean a permanent change in a DNA sequence between individuals within a population. So you can think of a population commonly having a certain DNA sequence, and then the variant will be something different. That's all it is. In the UK, we're sort of steering away from using the term mutation because it's sort of got a negative connotation. So it sounds nasty, basically. So they're sticking with the word variant. An inherited genetic variant occurs in germ cells, and that's sperm and egg, and a non-inherited variant could just occur in somatic cells, so that's other cells of your body. So when you hear the term variant, typically it's a neutral term, so it doesn't mean a variant is pathogenic or not. It just means it's neutral in terms of disease pathogenicity. So let's look at some simple variants. So what does a genetic variant actually look like? The sort of most common type you'll find in the genome is point substitutions. So at the top you can see substitutions where you've got one base, a C, changes to another base, a T, and that's called a point substitution. An insertion is just where you've got an extra sequence of DNA is inserted into a sequence. And a deletion is obviously just where some DNA is deleted and you've got a shortened length sequence. So how can these other ways to describe them? So this, in terms of coding point substitution, so if we're looking at substitutions in the coding region, that substitution could have different impacts to the protein. So because of the redundancy in codon usage, it means that a substitution could have no change to the protein because it still encodes the same amino acid. It could change the amino acid, and this could be a conservative change or a non-conservative change. And that just means the conservative change means you're swapping out for a similar amino acid, and the non-conservative means you switch into a different type of amino acid, maybe a basic to a polar. So it could have a big impact on the protein structure. The other impact of substitution could be, it could encode then a stop codon, which would sort of terminate the protein prematurely. So one of the things we notice is it's important that we describe a variant when we're publishing a paper with an accurate description. And because there's many different ways to describe it, that's where we see in the literature you get errors. So I'm just going to go over what's the best way to describe a variant. If you've got a paper and you're publishing your data, it's really important for clinical diagnostics because it allows accurate sharing and comparison of variant data. So the Human Gene Variation Society have a nomenclature system, HGMD, which is really well used, and we'd recommend you use that when you're describing variants. I'm just going to go into that a little bit more detail. So when you describe a variant, you can describe it at different levels. Obviously, you can describe it at the genomic level, you can describe it at the gene level, and the protein level. What we notice is when we're doing our curation for HGMD, about 15% of mutations published, there's some sort of error, or the mutation's not adequately described. So we can't determine where this mutation is. So that's why our advice would be at least describe a variant at two different levels. And so genomic level and gene level would be really good, or even gene level and protein level, but not just one of those. If you just include say protein level, it's going to be hard for, or even impossible for us to work out where the mutation exists at the genomic level. You may have heard of reference sequences. So this is before we had the genome in 2000. We already had these reference sequences for genes. They start with NCBI, they start with an NM_, and we can describe a variant in reference to these reference sequences. So it's always useful to, rather than just provide a gene name, provide a reference sequence ID. And then after that you can use a prefix like a C dot or a G dot or a P dot to sort of indicate what sequence level you're describing the mutation at. You can use ensemble, and that's a good set of transcripts to use, but at HGMD we use the represent. This is just an example of one. You will have seen these type of files. They're easy to work with computation as well. This is just examples of how you can describe various different types. You will see these in the literature when you're dealing with mutations. Typically when you're dealing with NGS, it comes in a different format though. You'd be looking at VCF format, which I'm going to deal with shortly. is really useful if you're dealing with mutations, because it can convert between these different descriptions, and it can take, if you give a description against a transcript, it can then map it back to a genomic location. So we find it really useful, and it's one of the best tools we've come across. So this workshop is about NGS, so next generation sequencing. And when you're working with next generation sequencing data, typically it will come off as VCF format, which is basically like a text file, tab delimited, with the genomic locations of mutations. It's really useful to work with if you're doing anything computationally, because you know exactly where the mutation is. So when you're using the HGPS nomenclature, often you have to map back to this VCF format to be able to work with the data if you're doing sort of a genomic type of analysis. So we just looked at how to describe genetic variation, because that's really important. If we don't describe it accurately, then we're not going to be able to use that data and share that data. But taking that further, how do we then prioritize variants which may be disease causing? So when you do, say you're looking at an exome screen, you're going to find a lot of variants that just come up. So you might be like 30,000 variants, and your job might be to identify the disease-causing variants from this 30,000. So there's a lot of different ways you can do that, but you can see that it's a bit like a needle in a haystack. So we've got to have a good strategy for narrowing down that set. So one of the ideas I'd like you to think about is that all variants, when you're looking at this 30,000, all variants have the potential, either on their own or in combination with other variants, to be disease-causing. So what you can take from that is you can use variant types as one of your first stages of prioritizing and filtering. So when you're developing an NGS pipeline to filter and prioritize this sort of 30,000 variants you've found, you're trying not to filter out the disease-causing variants, but you may still do that, so you just need to find this balance. So we would use mutation types as a rough filter. So this filtering, it doesn't mean that it's about probabilities, like where's the probability that your disease-causing mutation is. So if we can think that a mutation which truncates a protein or shifts the reading frame is more likely to be disease-causing than a mutation that doesn't impact the coding sequence. But also what we need to take from this, it doesn't mean that the non-coding variants cannot be disease-causing. So it's probabilities, and it's using these probabilities and these mutation types as a rough filter. It doesn't always work. So in line with this, so when we see a silent substitution and we think, oh, it doesn't change the protein sequence, that doesn't mean it can't be disease-causing. So that's another idea I'd like to get across. There's less probability that it's disease-causing, but it's not impossible. A lot of pipelines in NGS work with filter out silent mutations. So whilst they might not change the primary protein sequence via codon usage, they can change it via disrupting splicing, and also use of an alternative codon can alter the accuracy of mRNA translation. It can also alter the speed of translation, which is, if you've got a large protein like collagen, if you change the speed of translation, you can change the folding a little bit. we do see that silent variants are commonly under negative selection, so it means that they do have a function. So when we see around 2,000 silent variants currently reported as disease causing, so it's just something to be aware of. Don't just completely exclude them. Maybe you exclude them to start with, and then if you don't find any good candidate mutations, then you bring them back in. So definitely focus on the coding region when you're trying to identify disease-causing mutations. But I don't think we should be ignoring the non-coding regions. And Matthew Hayden, my colleague, is going to give us a bit more of an overview into that area, so I'm not going to go too deep on all of this bit. So with a non-coding variant, it's not clear what the impact is. When you're looking at a protein coding change, it's quite obvious. Like if you've got a nonsense mutation, you can say, well, this truncates this protein, and this protein is involved in the phenotype, so this is my candidate mutation. But when you've got a non-coding variant, it's more difficult to see the impact of that. You can't really, you need to use other tools to do that, and even though then, without experimental work, you haven't got much evidence. So I won't go into too much to how non-coding variants can impact things, but they can affect binding sites, they can affect, we can call structural variations, they could affect splicing, and they could affect regulation of genes up and down. We're trying to get an estimate of how much of the disease is due to non-coding, and because we're biased towards the coding region, that's hard to do. But based on a study looking at people with developmental disorders, they could only find so for 60% they could find, and 40% they couldn't find any mutation for these developmental disorders. They only looked in the exome. So I would say it's 10 to 40% currently of genetic disease is probably caused by non-coding, but that's just an estimate. Another complexity when we're sort of trying to analyse NGS data is to do with splicing. And what we've got is basically a gene encodes an mRNA, which is then spliced to form a mature mRNA that then is translated into a protein. And what this process allows is something called alternative splicing. So one gene can code for multiple proteins, and most genes in the genome are alternatively spliced. So that means that most genes code for more than one protein. So this alternative splicing also impacts how we interpret a variant, because if one gene makes multiple proteins, one variant could have an impact on one protein and no impact on another protein. So when we're analysing the impact of a mutation, we have to consider the protein that is most sort of biologically relevant to the disorder we're looking at. So in this example, we've got this 5 exon gene, and we can see that if we've got a mutation, protein A has all 5 exons. Protein B skips out exon 3. So if we've got a mutation in exon 3 that changes the protein sequence, in exon 3 and protein B, the impact of that mutation would be no impact potentially. So we can see that if we were analysing the impact of that mutation, we need to be careful with the protein that we're using to annotate it against. So in some cases we might use all three and say, you know, the impact in protein A is this, and B is this, and C is this. Another way is to look at what the most abundant protein is in that cell type and sort of use that to analyse the variant. So the take home from this is, it's important we pick the right transcript and the right protein when we're analysing a variant, because that can change your results. Alternative splicing does happen a lot in the genome, and these transcripts and the proteins that arise from them can be very different across different tissue types and developmental time spans. So you need to pick your transcript of interest carefully, and it may impact your results if you pick one that is not representative of the sort of disease you're studying. So I've sort of mentioned this already. So this is some evidence to support this. In one study they looked at about 300 genes for epilepsy, and they only considered one transcript. But 30% of these genes had sort of alternative splicing occurring, and they missed them because they didn't use the full range of transcripts. They missed 4 pathogenic variants, which were only found when they then reannotated with all the other transcripts. So good transcript coverage can improve clinical results. So we're going to move on now to looking at sort of variant interpretation and classification. So when you've got this exome, it is a bit like this needle in a haystack. And you might want to find this one mutation that's disease causing. But in practice, you're looking at probabilities. So you're sort of saying, I've narrowed down this 30,000 to 30 mutations that potentially could be disease causing. And you would then need further evidence to sort of narrow that list down. So it's not always that, you're not always able to just find one mutation that's disease causing without doing functional work. That's sort of the take from this part. So finding disease-causing mutations is very important. It can impact patient outcomes and it can impact what treatments they receive. So we need to get it right. This is something that's it's the foundation of sort of clinical diagnostics. I want you to be aware of there's these HGMD guidelines for variant classifications, and if you're working with mutation data in a disease setting, you'll need to be familiar with these guidelines, and they sort of divide mutations up into pathogenic, likely pathogenic, uncertain significance, which is a bus, and then likely benign, and they have 28 criteria to sort of assign a mutation or a variant into these categories. They use population data, in silico computational data, functional data, segregation data, de novo information, and other information as well, to sort of form these classifications. So they take each category and use that to sort of rank mutations. So you can sort of use this to sort of identify my pathogenic and likely pathogenic mutations from my exome. We're going to run through some of those now. There is some tools that can automate some of this classification, so you don't need to do it manually, but you won't be able to do the full classification automatically. But Intervar is a good tool to use to automate some of this classification. So we're going to just look into some of these types of evidence for pathogenicity. So population data, this is really useful. So you will have seen population studies such as EXAC or XAP is one of the big ones, and genomenon is the other one. They're useful for assessing the frequency of a variant in a general population, and this allows us to assess the pathogenicity of a variant. So what we're saying is, if you find a variant in your patient, and then you look at how common that variant is in the general population, you can then work out what is the sort of chance that this mutation could be the disease-causing mutation. Because if your variant is really common, it's unlikely to be disease-causing. This does depend on how prevalent the disease is. So if I see a mutation and it's at 30% in a population, and it's for a rare damaging disorder, it's not likely to be disease-causing. So we can use this information. So typically, we would exclude things that are at the 1% level. So I'm looking for rare variants. If it's over 1%, maybe it's not disease-causing. GenomeAD is a really good database for this sort of data. It's got exomes and whole genomes in it as well. It's probably the best data source to use. So if you're looking for population data, I would say head to this database. The next category of information would be in silico tools. So this is tools like SIF, CAD, Mutation Taster, and they will look using computational algorithms to measure the impact of that, or predict the impact of that mutation on protein structure, function, and other things. There's lots of these tools out there, but you've got to be careful. Just because a tool says a mutation is deleterious, it's just a prediction, so you've got to bear that in mind. They're not always correct. Often these tools use information such as evolutionary conservation, context within the protein sequence, and the biochemical consequence of amino acid substitution. Most of them use the same sort of feature set, so you might get the same results with different tools. So just because 10 tools tell you it's pathogenic, it doesn't add much weight because they're all using the same things to make the prediction. They're all going to tell you the same thing in general. So it's important to mix your tools. So you use two tools to make predictions, use tools that are different. That would be the take-home message. Really important data would be functional data. If you've got functional assays, that can really support whether a mutation is disease-causing or not. But obviously that is expensive in terms of time and money. But that is probably one of the gold standards. So there's lots of functional genomics technologies out there now that can look at data sort of in a high throughput way. If you've got access to this, it would make your job a lot easier. Another type of data could be family information, so segregation information. If you're trying to make a decision on whether a mutation is pathogenic or not, a segregation analysis is really useful. So what that basically does is, do all the people with the mutation have the disease at a high sort of level? And if you see that the mutation travels with the disease, and the mutation then, you say, segregates with the mutation, that means that there's a high chance that that mutation must be in both those people. It's got to be, basically. So you can use that information to help your pipeline, your decisions. So when a specific variant in the right gene that you're looking at segregates with the phenotype, especially if you see it in multiple affected family members, it's going to give you a good evidence of pathogenicity. The other type of information which you can do, if you've got parents, and you can sequence them as well, you can look for de novo variants. So de novo means it's not in the parents, and it's arisen de novo in the offspring. So if the parents haven't got the disorder, say it was autism, and the child has, and it could be a de novo mutation that is the cause. So you can then use that information as supporting evidence. But if the parents had the disorder, then you would tend to not be looking at de novo mutations. So databases, these population databases are very useful. You've got disease databases, which may have information on your variant, and also functional databases with assays about a given region of a genome. And you should leverage these databases to sort of provide supporting evidence for pathogenicity. Be careful when you're evaluating a database. Sometimes we just go for the free one, but it's important to look like how often is it updated, when was it last updated, are people using this source of data? Choose wisely, I suppose, but sometimes you've got to use what you've got available. So if you can only find this free database that's a bit old, you just got to go with that. But just try and assess the data quality if you can. So we sort of run a disease database ourselves. How could that be used to assess pathogenicity? So you take your variant that you found in your patient and you see, has it been reported before as causing a disease? If it has, that makes it a good candidate. If you didn't have a database, you'd have to go searching PubMed and try and find your variant, and that can be very time consuming. You can also use the disease databases to look at your gene of interest and sort of understand the mutational spectrum for that particular gene. So what we see is, for some genes, the disease is often caused by nonsense mutations, whereas for another disease, it might not be, it's more missense mutations. So if you understand the gene in question, disease-causing mutations that are observed, you can then sort of use that information to help guide your filtering. Other disease databases is ClinVar, which is a good resource for free one. Sometimes they sort of got, in terms of these categories, they may have a variant it's in pathogenic and benign, so that can be a bit difficult to work with, but it's a good alternative if you can't afford to pay for a resource. At HGMD, so we only collect germline mutations, so that's inherited mutations. We've got around, it's 400,000 now mutations for about 15,000 disease-causing genes. And we manually curate this data. We do have a free site, and we have a paid version as well. The free version is just out of date by 3 years, but you can still register for that and access the data. We use different disease categories, so we have DM, which would be disease causing, and DM question mark, likely disease causing. We don't collect mutations that are benign or likely benign. When we do a comparison of HGMD against other disease databases, we can see that we have we cover everything that's in OMIM, and we have a large overlap with ClinVar. And ClinVar only captures about 24%, 25% of HGMD. So it's probably useful to use multiple resources so you've got full coverage. So the key points is when you're doing clinical diagnostics and you've got this full exome of lots of mutations, identifying a single variant responsible for disease is tricky. And you might not be able to narrow it down to one. You might just be able to narrow it down to a set of mutations or variants. And that's about prioritizing and probabilities. And when you're talking to a clinician, you have to explain to them that it's probable that the variant is in this set, but it doesn't mean that it's definite. It's a probability, high probability, yes, but the disease-causing variant could be still have been filtered out. Based on these probabilities again, it's more likely that your variant will be in the coding region, and that's obvious. But if you can't find a variant in the coding region that's likely, you need to expand your search to include the non-coding regions. And that's something that we've seen a shift towards now, and it will sort of increase. We'll be able to sort of analyse the non-coding regions better, I think, in future years. And then if you do use in silico tools, they are very useful to help make, to sort of filter your data, but remember that they're predictions only. And if you're giving results back to a patient, and if there's a clinical consequence, you should use this sort of, the ACMG's framework, which relies on multiple lines of evidence. And that's the important thing, multiple lines of evidence. Don't just use in silico tools, you've got to use the full set. So in silico tools, population data, segregation data if you've got it, if you've got functional data, that's really good. But it's just showing that you can't just rely on one thing, basically. And I'd like to acknowledge everyone at HGMD. We've got a really good team, and everyone's supporting us working on this project. So it's, I've got to acknowledge their contributions as well. Thank you.
Thanks, Matt, for this nice, excellent presentation. So, I have a couple of questions. This Matt Hayden, are you going to present now or after the question?
I'm quite happy to do some questions, but
Okay. So there are one or two quick questions. Can I have the legend? Can I have that one? One question is that, Matt, I think you mentioned that the audit pricing and appearance of those audit slice products in the different parts of the cell. So is there any correlation between the two? Like one kind of audit splicing is occurring in one type of cell or one type of organ or something like that?
Yeah, that happens regular. So you're looking at a brain disorder, right? A brain cell might have one variant expressed highly and the other one very low level or no level. But if you move to a different cell type, it can be completely different. So the different transcripts are expressed at a high level. So that's why knowing something about the disease you're looking at and the cells that it impacts can help guide your decision on which transcripts you use to analyze it.
Yes, the next question is, while analyzing genome data for mutation through various online tools, do the algorithms take alternative splicing in consideration, or is there any tool to predict results based on alternative splicing only?
That's a good question. So a lot of the tools, if you put in a VCF, they will annotate against multiple transcripts. And then I think now a lot of them do like that. It will include all transcripts. It will include predictions based on different transcripts. So what that means is for that variant, you might get 5 predictions scored. So then some people say I take the highest score and use that, you know, if I get the highest score, but like we're saying, if that transcript is not expressed at a high level in myself like I'm interested in, maybe I should use a specific one. So that would be a good one. CAD, not sure if CAD, I like to use CAD, but I'm not sure if it uses, it would produce different scores for different transcripts. I think it just produces the highest score.
There is another question. Generally, missense mutations are responsible for causing disease, but the nonsense mutations don't affect the protein function. Is there any probability that changes it into the nucleotide positions? Right? However, however, it codes for.
However, it codes the same protein will affect the function.
So is it clear to you, Matt, or should I repeat?
Just say the last bit again.
Generally, missense mutations are responsible for causing diseases, but the nonsense mutation don't affect the protein function. Is there any probability that changes in the nucleotide position codes in the same protein will affect the function?
So what was the middle program?
So, if there is a missense mutation, sir, if there is a
Yeah.
nonsense mutation, that the effect is not seen in the protein function, right? So, can we predict?
So I say that like missense mutation could have a milder impact on the protein function, but nonsense still can impact protein function by truncating.
Is that the question, or do you want to ask something else? Yeah, I think it's a continuation of the previous one. Yeah.
Sir, actually, if we see changes in the nucleotide sequence first, second, or third position, but it code for the same protein, is there any possibility that it can affect the protein function or it has some deleterious effect?
So are you saying that looking at the triplet that took amino acid, and you asked about position 1, 2, and 3, is that right? So all of those positions have an impact on the protein because by splicing. So even if you get a change in that codon that doesn't change the amino acid, it can disrupt splicing by the tonic splicing regulatory element, which can mean that an exon is skipped. So you can have a silent mutation, skipping an exon, and leading to a different protein product, different sequence. So yes, every position could have an impact, but some more than others. So that probability, a silent mutation is less likely to.
Yeah, I think that answers your question. So for Dr. Matthew Mort, we don't have any more questions. So thank you very much, Mort. Are you staying online with us, right, Matt?
Yeah, I'm going to stay online to listen to the article, and if you've got any more questions, that's the way.
Okay. Thanks, Matt. So now we are going to have Mr. Matthew Heath, and he's an experienced bioinformatician in HGMD in Cardiff University, and he will be continuing from where Dr. Matthew Mort left. So over to you, Matthew.
Hopefully you can see my slides. So I'm Matthew Heath, I'm also from Cardiff University, and I work at HGMD and MAT. I'm the junior mat at HGMD, and both Matt and I are quite interested in the non-coding genome. And Popular Science has named for this, they've called it dramatically the dark genome. So that's going to be my talk today as the dark genome. So the contents of this talk, first of all, I'll introduce the concept of the dark genome. Then I'll talk about regulatory elements, so talk on the genome junkyard and non-coding RNA.
Matt, I think your slides are not coming up.
Oh. Sorry, just check. Matt, Mort, can you see the slides? Or?
I can see the slides on the trip.
So the non-coding regions that Dr. Mort just introduced, it's something that's of interest to myself and Dr. Mort, and we get rather annoyed when people ignore it because it is such a huge part of the genome. So the talk is in 4 parts. I'm going to introduce the concept of the dark genome, regulatory elements, then I'm going to talk about something I call the genome junkyard, and then non-coding RNA. So that's the first part is my introduction. I'm going to talk about the human genome, encode, and the junk or function argument. So there's a quote here from a BBC online article. It says, if you look at genome-wide association studies, which search for genetic variations in large numbers of people to identify those linked to disease, the vast majority linked to chronic illnesses like Alzheimer's, diabetes, and heart disease are not in protein-coding regions, but in this dark genome. So the Human Genome Sequencing Project was finished about 23 years ago, it seems like yesterday. During this Human Genome Sequencing Project, it was expected to find about between 25,000 and 40,000 genes coding for proteins. However, we now know that there are about 19,000 genes making up the coding region of the genome. So what percentage of the entire genome is this? So it's about 1 to 3% of the genome is protein coding, that's tiny. The rest is referred to as non-coding DNA, as in non-protein coding. And this is what popular science journals and articles refer to as the dark genome. So there's another quotation here. In spite of the significant improvements in molecular diagnostic rates for rare genetic diseases resulting from implementation of whole exome sequencing, the molecular diagnostic rates have leveled off approximately 30 to 40%. The reasons for this are many, but are likely to include mutations in 98% of the genome not covered by whole exome sequencing. So returning to this pie chart, you will see lots of variations on this pie chart on the internet. It's a useful guide and introduction, but it's a bit incomplete. It doesn't note, for example, that around about 80% of the genome is actually transcribed. That means it produces RNA. 20 to 30% feature regulatory elements in the genome. And pseudogenes take up 1 to 2%. it also doesn't illustrate very well that there can be overlap here. For example, you can see lines and signs, but you can actually have lines and signs coded for in the intron. You can actually have some protein coding genes in large introns. So a sort of one-dimensional pie chart doesn't really tell you the whole story. Final quote, I promise. The problem with drugging proteins is that there are only 20,000 or so proteins in your body, and most of them are expressed in many different cells and pathways, which are unrelated to the disease. The dark genome is exclusively specific in its activity. There are non-coding RNAs which regulate fibrosis only in the heart, so by drugging them, you have potentially a very safe medicine. So, going to talk briefly about ENCODE. This was a project started around about 2012. It was considered the next big follow-up to the Human Genome Project. And it's the encyclopedia of DNA elements. It was a collaborative project and it's aimed at identifying and characterizing functional elements within human and other selected genomes. Now, it focused on regions of the genome that are involved in gene regulation. This is promoters, which you've probably heard of, but also things like enhancers and non-coding RNAs. And there's a link to the project main site there. So I'll go over this diagram in more detail in a moment, but to give you an oversight of what's included in ENCODE, it contains data on DNA methylation and long-range chromatin interactions, such as the looping, and both of those affect transcription. It also annotates the binding activity of transcription factor proteins, which I'll talk about in a second, and the architecture of this gene regulatory elements, such as the promoter region, and some of these very long-range regulatory elements. I'll discuss that more in a second. And just to show you how useful the information in ENCODE is, and if you aren't aware of it, it's worth checking out. It's open source, it's free, and it enables researchers to explore these complex regulatory landscapes in the genome, in this dark genome. So you can gain insights into the mechanisms and various biological processes that go on in the dark genome. And you can see here from this graph that the number of publications involving ENCODE data is increased dramatically since about 2013. And I just wanted to talk about this idea that you probably all have. You're a bit younger than me, so maybe not so much, but it used to be considered that this dark genome, this non-protein coding region of the gene, is just junk, rubbish. As far back as the 1960s, they sort of calculated that around about 10% would be protein coding. Obviously, they overestimated it. And they thought the rest just wasn't useful, it was just evolutionary rubbish that was left behind. However, this idea was a lot more widespread in popular science than it was in proper science. Most geneticists were aware that dark genome was not junk, because evolution doesn't consider junk readily. And I should also say you can be a bit critical of popular science and sometimes the things it says that are slightly incorrect. But this sort of thing gets into the public imagination, and that's where research quite small. So moving on from junk, ENCODE came along and they said, hang on, it's not junk. We think that about 80% of the human genome has a biochemical function. This relates back to what I told you about 80% of the genome being transcribed. Then other people said ENCODE is setting the bar slightly too low in suggesting function just because something is transcribed. And they said there are regions of the genome that might be transcribed, but they're not actually useful to the host, and these transcripts are broken down. So we have a sort of compromise going on at the moment. Not all biochemical activity necessarily benefits the genome's owner. Junk can also be useful. I'll talk about that more in a moment, but one brief idea why junk DNA might be useful is it actually spaces out the important coding regions of the DNA. I've illustrated this with a knife throwing here, and just to show that when he's throwing the knife, if you imagine the lady in question as being functional and the board as being non-functional, then by the law of probabilities, you've got a fair chance there of hitting the board instead of the woman, hopefully. And what this shows is if you have damage, say UV damage to the genome, if it's spaced out with junk, then there's a chance that that junk will take the hit, and the variant E coding region will be protected by the fact, again, a lot of probabilities, that there's just as much chance of hitting this junk. In fact, quite a lot more so if you consider how small the protein coding regions. So the second part, I'm going to talk about the regulatory elements that I just mentioned. So the short version of this, if you fall asleep, is that regulatory regions are stretches of DNA that proteins can bind to. This binding and unbinding helps run the genome, like clockwork. So, going back to the diagram I showed you earlier, this is unraveling the chromosome, which you can see in the lower right. And the chromosome consists of these things called nucleosomes. Now, nucleosomes are histone proteins that have DNA wrapped around them. And these are all bunched together and all wrapped up very tightly, and that's what the chromosome is. And to get to the DNA, you obviously have to unwrap the structure. And this is about condensation and control. And what do I mean by this? I don't know if you can see, but I have a very expensive prop here, just a reel of cotton. Obviously, if you just jam cotton in the drawer when you finish with it, it'll get all tangled up and damaged. However, if you put it in a reel, it's nice and safe, and it stores it, it doesn't get damaged. But control is another function. If you think about transcription factors and how DNA is transcribed and translated, well, if it's tightly wrapped up, you can't actually get to the DNA to transcribe it. So this is a way of controlling transcription in the genome, just by hiding all the DNA away and wrapping it up. Quite expensive problem. So genes are not expressed unless they're accessible by RNA polymerase and transcription factors. So chromatin must open up in order for this to take place. And this process is known as chromatin remodeling. So I've just spoken about transcription factors and how important it is for them to actually have access to the genome for anything to happen. So what are they? They're proteins that regulate gene expression by controlling the transcription process, which is the synthesis of RNA from DNA. Now they can activate or they can repress gene expression. Activators obviously enhance transcription by promoting the binding of RNA I can't say that with this one. RNA polymerase to the gene's promoter region, or repressors, obviously, inhibit transcription. So they have crucial roles in various cellular processes, including development, differentiation, response to environmental cues, and maintaining cellular homeostasis. I think there was a question earlier on various alternative transcripts. Well, that involves certain transcription factors. You'll have different transcription factors activating different transcripts at different times. It's very important to remember that you have between about 50 and 100 trillion cells in your body, and those cells are differentiated into about 200 different types. So you have a very, very busy genome. This is unwrapping, rewrapping, transcription factors coming and going. It's incredibly busy all the time. So I'll just talk about some of those regulatory elements. You probably know about promoters from basic genetics. These are the binding sites for the protein machinery that carries out the transcription, and it's usually just ahead of the gene. Enhancers are binding sites for transcription factors that help activate transcription. Now, they can be found on the DNA strand before or after the gene they control, and some of them can actually be found quite a way away from the gene, outside the gene. I think there's some that are on different chromosomes to the gene. Silencers, binding sites for transcription factors that repress transcription. Important to remember that these transcription factors are turning on and turning off as well. Insulators, binding sites for transcription factors such as enhancers, blocking the insulators that prevents enhancers from aiding in transcription. So they block the thing that helps the thing. And you have barrier insulators prevent structural changes in DNA that repress gene activity. So these structural changes would stop transcription. These barrier insulators go in and stop it. So it's quite a complicated thing. I've tried to illustrate it here, I think perhaps this slide as well, to show you the transcription machinery and how it's not just this polymerase that comes in. It's this huge machinery involving transcription factors. So you've got these activators, you've got these enhancers, and you've got a promoter and a polymerase forming this machinery. Something I haven't spoken about because they can be all one talk is methylation, and that's a way of coating the gene to shut it up and stop the transcription. Briefly, some databases. Jasper is an open source database, contains manually curated non-redundant transcription factor binding profiles for these transcription factors across these taxonomic groups. Transfacts, another version, has got public and free versions. And it's just a database of these transcription factors and how tolerant they are to change. So this might be a good resource if you're interested in investigating mutations and transcription factors. The third part of the talk, I've called this the genome junkyard. In this, I want to briefly go over transposable elements, pseudogenes, and introns. So again, the short version, transposable elements are selfish genetic fragments descended from viruses, or some of them descended from viruses, and they live in our DNA. Pseudogenes were originally considered once functional genes that are now old mutated rats in the genome. And